Hi and welcome to my OCR A-Level Biology revision with me, Christine. So today's lesson I want to look at the light independent reaction. So this is part of your photosynthesis module in module 5. So let's just remind ourselves about the light dependent reaction because this is necessary for us to move on to this light independent reaction. So we know in non-cyclic photophosphorylation that photons of light are absorbed by the photosynthetic pigments in our photosystems. Our photosystems are then able to donate electrons from the reaction center from the primary pigment chlorophyll A. Those electrons get passed through the electron transport chain, and that is going to pump hydrogen ions into our thylakoid space. The electrons that are donated from our photosystem 1 will move up to allow for the reduction of NADP, a coenzyme. So that's the first product that we're interested in from the light dependent reaction, which is needed to move on to our light independent reaction. The next product we're interested in is the fact that we're going to be pumping these hydrogen ions into the thylakoid space. They are going to accumulate and then they are going to diffuse down their proton gradient. And when they diffuse down that proton gradient through ATP synthase, that's going to provide the necessary motive force to allow for ADP and inorganic phosphate to be joined together to form ATP. So that is another product from the light dependent reaction which is necessary for our light independent reaction. So our light independent reaction is also known as the Calvin cycle. It's called the light independent reaction because it does not require light. It occurs in the stroma. What it does require are the products from the light dependent reaction. If the products from the light dependent reaction are not present, then the light independent reaction will slow down and then cease to continue. So let's have a look at this light independent reaction then. So the first thing we need to understand with the light independent reaction is that we are going to be using the products from the light dependent reaction. So I've put them up here in my top left hand corner just to remind ourselves where these come from. But the first thing we need to understand is that we have some molecules. The first one is a 5-carbon molecule which is called ribulose biphosphate. Now you need to understand that that is R, capital R, small u, capital B, capital P. So you can write it as RuBP, but this is a ribulose, a 5-carbon pentose sugar biphosphate. Now, it's biphosphate because it has 2 phosphates attached. When we have that RuBP and carbon dioxide come together, what we have is a fixation of the carbon dioxide. Now, what you can see here is I actually have a enzyme called RuBisco. So RuBisco is ribulose biphosphate carboxylase. It is an enzyme that is going to fix carbon dioxide into this molecule. So we've gone from a 5-carbon molecule to now having a 6-carbon molecule. But this 6-carbon molecule is very unstable and automatically will lyse itself into 2 3-carbon molecules. Now you do not need to know the name of the 6-carbon molecule, but you do need to know that we call these 3-carbon molecules GP. Now another way of saying this is glycerate 3-phosphate. So our glycerate 3-phosphate is what we call GP. So in your exam, you only need to write it as GP. What will then happen is after the carbon dioxide has been fixed into the GP molecules, is we will then use ATP and the reduced NADP to reduce the GP into triose phosphate, TP. So we're converting our GP into a TP molecule by using the products that came from the light dependent reaction. So if, for example, there is no light, that would result in us not being able to convert our GP into TP. Well, now we have our triose phosphate. We have our 3-carbon molecule. One of those triose phosphates will go out of the cycle, and that can be used to combine together to form many different molecules, and we'll look at that a bit later on. That therefore means that we now have more TP molecules that need to be converted back into our RuBP. So what we need to do is we need to take more product, ATP, from the light dependent reaction, and we use that to do what's known as regenerating our RuBP. Now, remember, one of those triose phosphates went off to become another molecule. So to ensure that we are able to regenerate RuBP, we actually have 5 TPs that are used to regenerate our RuBP. So as it goes around the Calvin cycle as a whole, you actually start with 3 RuBPs, 3 carbon dioxide, you then produce 6 GPs, you therefore need 6 ATP molecules, you need 6 reduced NADP molecules, and you get 6 TP molecules. So it's important to remember that as you are going around, one cycle would only regenerate 1 RuBP. So therefore, it has to happen in a number of cycles to ensure we end up with this regeneration. So let's just recap that then. When we go from our RuBP and we use Rubisco to accelerate the joining together, the fixation of the carbon dioxide, we are actually using an enzyme which is lowering that activation energy, and that is going to result in this 6-carbon molecule which is unstable. That will then split into those 2 3-carbon molecules, which is our glycerate 3-phosphate, which you call GP. We then use 1 ATP molecule per GP molecule to convert it into the triose phosphate, but it also needs to be reduced. So the reduced NADP needs to be oxidized. So it's important that you understand, for one substance to be reduced, the other has to be oxidized. So when we are reducing our GP to our TP molecule, our NADP has now been oxidized. So we are adding the hydrogens in to finish that conversion of our GP to our TP molecule. So therefore, we need to utilize those products from that light dependent reaction. The last part then, to be able to continue with this process, because it's a cycle, the Calvin cycle, our RuBP, which is used to fix that carbon dioxide, must be replaced. So therefore, you require 5 of those TP molecules to regenerate the RuBP. And because we are rearranging the atoms, ATP is required to provide the necessary energy, which is going to allow for that conversion. So it is important that we remember that the Calvin cycle is always these 3 parts: carbon fixation, reduction, GP to TP, and regeneration of our RuBP. So let's just have a think then, what happens to that triose phosphate, that one TP molecule that has gone off and has left the cycle? Well, that TP molecule could be converted into a glucose phosphate. That glucose phosphate could then be added to fructose phosphate and create our sucrose. Now that will link to the module 3 topic area when you do transport in plants and you look at translocation. So the glucose that's been produced by photosynthesis in the light independent reaction could go off, be added to fructose to produce sucrose to then be transported around the plant to where it needs to go. The glucose phosphate could actually be used in respiration. It could be used in glycolysis to break down into the triose phosphate again, therefore providing 2 ATP molecules that may be necessary for the cell to function. Or what could happen is that glucose phosphate could be stored as a starch polysaccharide, or it could be converted into a beta glucose and stored to make cell walls, the cellulose polysaccharide, which makes up your cell walls. So there are different ways in which the plant can use the triose phosphate that has been produced by that light independent reaction. It could be converted into fatty acids. So fatty acids could be synthesized and therefore you end up with your oils in your plants, or it could be converted into amino acids. Amino acids can therefore then be joined together in protein synthesis through the process of translation, resulting in the growth of the plant. So it's important that we understand the light independent reaction requires the products of the light dependent reaction and the presence of carbon dioxide. And I'll do a whole video on limiting factors that will affect the rate of photosynthesis. I hope you have liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you have not done so already, please do check out my revision platform, www.aiqchat.com.